Alrighty, this is the last part for chapter 1 of the Pierce textbook, Introduction to Genetics. And what we're going to talk about in this portion of the lecture is some genetic concepts. I cannot stress enough how important it is for you guys to have a really good handle on what these terms are and what they mean. So first, let's talk in a broader sense, total cells. So eukaryotic and prokaryotic cells. I'm sure you guys have heard that terminology before. I really hope that you know what they are and how they're different. But in short, eukaryotic cells are very, very complex. They've got sophisticated organelles and systems in place that help them to function regularly. So they're going to have things like nuclei, Golgi body, um, mitochondria, actin, all of that good stuff. And so we see eukaryotic cells both in animal and plant cells. A simpler type of cell that research says we evolved from is prokaryotic cells. So very, very simple. They have just a cell wall, a plasma membrane, and then contained within that cell area is cytoplasm that has ribosomes in it, and the DNA material is contained in the center of the cell but is continuous. So in prokaryotic cells, they tend to only have one chromosome. It's pretty large, but they only have one chromosome, whereas in eukaryotic cells, we have multiple chromosomes. So just for example, humans have a total of 46 chromosomes, and cattle have a total of 60 chromosomes. So much more genetic material and more complex cells. So the big difference between our eukaryotic and prokaryotics is our more sophisticated eukaryotic cells have many different organelles. And the one that we're going to be the most concerned with in the front portion of this class is the nucleus. And that's because that nucleus contains the DNA, or deoxyribonucleic acid. And those acids are going to be strung together in a very specific sequence, and they're going to code for genes and these genes are going to reside on chromosomes. So the nucleus is very, very important because it actually works as a gatekeeper. So it controls access to the DNA. So it is going to only permit certain proteins to get into the nucleus and certain proteins to get out of the nucleus. And this really is an integral part of the sophisticated procedure of the cell because it's going to keep DNA separate from the metabolic machinery. So DNA, in its most simplest terms, is the blueprint for every form and function. And DNA is going to be made up of chromosomes. So we can see those chromosomes are going to be stored in the nucleus, and chromosomes in the traditional sense are kind of like in X and Y configurations. So DNA structure is a double helix that is going to be linked together by hydrogen bonds. So we have one strand of DNA is going to be bonded via hydrogen to the second strand of DNA. So DNA is going to be built from subunits called nucleotides. We can see those, a single unit of those right here. And each nucleotide is composed of three major components. So a sugar, a phosphate group, and a nitrogen base. So our nitrogen base is going to be in blue here. That's our adenine, guanine, cytosine, or thymines. And then the phosphate group, or the sugar phosphate backbone, is going to be the red, that's the sugar, and the P is the phosphate here. So this creates a backbone. So again, the four nucleotides that make up DNA are thymine, cytosine, adenine, and guanine, or T, C, A, and G. And their structure, you can see, is a little bit different, but for the most part, they all have the same major components. So these four nucleotides are going to be come together in the form of DNA and make up a very precise sequence. So nucleotides are going to vary specifically. So adenine is always going to pair with thiamine, or A with T, and guanine is always going to pair with cytosine, or G with C. So as you can see here, we have A's and T's always pairing together, and C's and G's always pairing together. Thiamine will never bond with guanine, and adenine will never bond with cytosine. So in order to make DNA divide correctly, it has to go through a reproductive process. So every time a new cell divides, or in this case a somatic cell, that is going to be termed mitosis. And mitosis is the simplest process, and it is actually contained within the process of meiosis as well. And that'll become apparent when we get to chapter 2. So in order for cellular reproduction to take place so we can duplicate the DNA, the double helix is going to have to unwind, and we're going to start synthesizing a new strand of DNA against this backbone of the older DNA. So the new template is going to then bond to the old template. And this process is called DNA replication, and we're going to study it very in depth in chapter 12. So just to give you an idea of the size of genomes among many of our animal and plant species, the size of the genome can be interpreted as the total number of chromosomes for each species. So we all know humans have a total of 46 chromosomes, 23 pairs. Cattle have 60, swine have 38, and the dog, man's best friend, has 78 chromosomes. So very big genome. On the plant side, we have really huge genomes here as well. So we've got the sweet potato and sugarcane that has a lot of chromosomes, 80 and 90 respectively. And then the smallest one looks like it's going to be the banana here, and it only has 33 chromosomes. So that becomes very important because the banana is actually really at risk for becoming extinct because we have bred it for so long to be resistant to certain diseases, but we didn't anticipate new diseases moving in. So we've created this genetically similar organism in the form of the banana that doesn't have any genetic diversity to deal with new extraneous and environmental factors. And so genetic diversity is directly related to how big the or how many chromosomes an individual species has. So DNA, of course, is the language of life. So this is going to be common between all plants and animals on Earth. So as we look at the total, or how DNA is, or how basic DNA gets, we've got our entire plant. So if we were to look in the leaf of that plant, we would see these chloroplast cells. And if we were to look in one of the chloroplast cells, we would see the chromosome in the center. Within that chromosome, we have DNA, and that DNA is going to be broken down even further into the 3 nucleotides, A, G, C, and T. And so there's a lot of information carried in this step-by-step process. So our DNA is going to be stored in chromosomes, and chromosomes is going to be the double helix form of that DNA. So this becomes very important because as it houses DNA, it, along with histones, house DNA, we can actually go through very specific procedures to look at the chromosomes that are going to be associated with an individual. So for example, this is a karyotype that was taken from amniotic fluid of a fetus that was about 5 months in gestation, and we can see that through a very specific process and lab technique we were able to isolate all 23 pairs of chromosomes, and we know that this was a boy as he has an X and a Y chromosome. All right, so let's make sure that we get some terminology down, and this is a really great place for you guys to do flashcards or Quizlet cards or whatever you need to do. So on the left-hand side here, we have 2 chromosomes, the blue one and the purple one. So these chromosomes are paired up together, therefore they are going to be homologous. So homologous meaning the same. Okay, so one is going to come from mom, one is going to come from dad, but they still are going to be the same chromosome for an individual. So the red areas that we see here at the top portion of this chromosome, this is because we're looking at the same area of each chromosome. This is known as a gene or a locus. We also may call these loci. And this refers to a site, a very specific site of a gene on a chromosome. As we break that down a little bit further and look beyond just a gene or a loci, that gene and its association with either the chromosome coming from dad or the chromosome coming to mom is going to be referred to as an allele or an alternate form of a gene. And then, so what we see down here at the bottom is going to be pairs of genes. So the red pair, the purple pair, and the orange pair that we see here. So two alleles are going to come together and they're going to form a pair of genes. All right, so a gene, very specifically, is going to be a segment of DNA that codes for a specific trait. So this could be for coat color, or it could be for plant height. Okay, so it has a very specific trait that it's coding for. Contained within that gene are going to be certain pieces of anatomy associated with the gene. First of all, the gene is going to start with a promoter region, as shown here in orange. The promoter region contains a start site, which is usually over here to the left, and this is going to be the site that the gene is going to be signaled or be turned on. So once the start site is activated, then we will get transcription and then translation to take place. After the promoter region, we have a series of exons and introns. Exons, or the pink portion showed here, are going to be coding regions. So these are going to be regions of the gene that are actually transcribed into messenger RNA. So from DNA to RNA, also known as the process of transcription. The second area, the yellow and the white here, these are going to be introns, and these are non-coding regions, or what we used to term as junk DNA. Now, just as a side note, we're seeing lots of function that has to do with what those areas that we thought once were introns. All right, so the central dogma of protein expression really comes down to genes are going to be used to code for protein. The protein is actually going to be the medium that causes the effect, so the increase in plant height or increase in body weight. So proteins express the genetic trait, okay, whereas DNA carries it from generation to generation. So the way that we synthesize protein or get genes to be expressed is a multiple part process. First of all, in the nucleus of the cell, we're going to produce messenger RNA. That messenger RNA is then going to travel outside of the nucleus of the cell to the Golgi apparatus and then into protein to be packaged. So multiple step process that actually requires multiple parts of the cell.